Eyes are incredibly sophisticated, beautiful organs. We humans can detect 8 million colors. We can see both day and night. Some animals can't do that. We can focus our eyes from a single hair to the horizon in milliseconds, automatically. The furthest our eyes can see is the Andromeda galaxy, which is 2.5 million light years away. That's far. We see in 4 steps. There's a stimulus, like this fishbowl, washed by light, and that light is reflected from the bowl to our eyes. Our eyes create an image of the stimulus on the retina. There are receptors in the retina that transforms the light into electricity, and in the last step, that electrical impulse goes to our brain and is processed, and seeing completed. There is this incredibly weird disease on seeing that I want to share with you guys. Our eyes work contralateral. Whatever is in your right goes to your left hemisphere, and everything on your left goes to the right hemisphere of your brain. And the bundle of fibers, the bridge which connects the left and right brain, is called the corpus callosum. This incredibly weird thing happens when a person's corpus callosum, this bridge, severed. It is called a split brain. There is this test. Only look at the center. Don't move your eyes. What did you see? A burger. When you perform this test to split brain patients, they give the right answer, so no problem with seeing that. Let's do it again. You saw the elephant? Patients give right answer. They see the elephant too, so they can see their right visual field. Let's do it one more. What did you see? A sunflower? Split brain patients can't answer this one, and they say, I didn't see anything. So they can't see things when they are in their left visual field. But the amazing thing is, when we give them paper and pen and ask them, just draw me something that pops into your mind, something you might have seen, they draw this. How does this happen? How can they draw something that they couldn't see? We will solve this puzzle, but first, let's see the anatomy of the eye. There are 3 types of muscles around our eyes. These muscles control upward, side to side, and downward movement. Eyeball has 3 layers. The first one, the outermost, is the sclera. It is the white of our eyes. It is a tough, hard material and is around 1 millimeter in thickness, which makes it thicker than the other 2 layers. At the very front of the eye, this outer coat loses its white coloring and becomes transparent. This is the cornea. If it wasn't transparent, it couldn't allow any light to enter iris, which is a muscle that controls how much light enters the eye, and it gives our eyes their characteristic colors. You probably know this, but this is the pupil. The pupil is where the light enters our eyes. It's actually an opening or gap inside our eyes, and it is the darkest color we have in our bodies, jet black. All the lights that illuminate the universe pass through this dark spot, but it cannot save the pupil from its darkness. And there is a lens behind it. It changes shape to focus light on the retina. Underneath the sclera, there is choroid, which nourishes the eyeball because it contains blood vessels. The innermost layer is the famous retina. Retina contains millions of light-detecting cells. It detects light and sends neural messages to the brain. Inside the eyeball, there is a fluid called vitreous humor. It's a gem-like substance, something like an egg white. Lastly, this is the fovea. It's a special spot on the retina. It's a classical question if you have a test tomorrow. And here's the answer. There are 2 types of cells in the retina, rods and cones, which are shaped like rods and cones. They are photoreceptors, so they detect light. They pick up electromagnetic energy. Rods work well in darkness. They only provide a black and white image, but they are incredible at low-level light. Rods are sensitive to brightness and movement. They have poor acuity, so they have low sharpness and focus. Rods are better at peripheral vision rather than direct. Cones are opposite. They are good at focusing in the center and bad at peripheral vision. They have excellent visual acuity. They provide sharp and clean images. Cones are sensitive to colors, and they can't be used at night. They are good in daylight. There are 19 times more rods than cones in our eyes. Cones give us this picture, and rods give us this one. Our brain put these two images together to have a better picture. And fovea is a special place for cones. There are almost no rods in the fovea. They are located anywhere except the fovea and blind spot. Cones are everywhere too, but they are concentrated in the fovea. Did you know that some scientists consider the eyes part of the brain? Our eyes are completely useless alone if it was not for the brain. There are 6 stages of seeing. First is reception. Rods and cones are picking up the light, which is electromagnetic energy. Then there is transduction. Electromagnetic energy is converted or transduced into electrochemical energy, so our neurons can use it or understand it. Now information sensed by the rods and cones can be sent as neural impulses to the brain. The next stage is transmission. Neural impulses are sent to visual cortex of the occipital lobe via the optic nerve. Then it is time for selection. Some features of the stimulus like lines, angles, edges, and some patterns are identified and discriminated against. The next stage is organization. Elements like features are grouped to form a whole, using perceptual principles, so it can mean something. The last stage of seeing is interpretation. The whole that has been created is given meaning. So, seeing happens in the brain, mostly in the occipital lobe. There are specialized parts of the brain for seeing, hearing, talking, and everything. About the split brain patients, remember Broca's and Wernicke's area? I made a video about that, but shortly, understanding language and producing speech happens in Broca's and Wernicke's areas, and they are both in the left hemisphere, so we communicate with our lefts. When an image flashed in our right, we see it with our left brain, and even if you have a split brain, you can say what it is. You don't need the corpus callosum, since the electrical signals are not needed to transfer to speech center. But when I show you a picture which is on your left, it goes to right hemisphere, and your brain needs to take that information, the picture of the sunflower, and carry it from right to left to the speech center. If your corpus callosum doesn't work, you can't articulate what you had seen. But you can draw, why not? So, isn't it interesting on so many levels? Can you be sure of what you perceive? Sometimes you think you had seen something, but you were tricked. Sometimes you say you don't see anything, but you do. As Edgar Allan Poe once said, believe nothing you hear, and only one half that you see.